Hello and welcome to this week's What Were They Thinking? Among the variety of news items we have for you this week, there is a development in regards to antidepressants and treatment for brain cancer, and thankfully based on a relatively readily available and already approved antidepressant. The relationship between diabetes and bacterial infection, and specifically type 1 diabetes. The increasing rate of breast cancer diagnoses, but decreasing rate of mortality. The best, or perhaps we should say worst, choice of thickness for paper cuts, and the use of this knowledge is going to be up to you. Retraction, or at least extreme criticisms, of a paper looking at external key, and the bizarreness of it. Why it is that although humans have bigger brains, it was not without cost, and lastly, the idiocracy, or at least what was idiocracy, has become fact where police raid a radiology facility and encounter the obvious result of being near an MRI with metal objects. The timestamps for these and other news items can be found in the description box below. Let's start with treating brain cancer, and not the usual brain rot like internet, but actual cancer. One of the most nasty and aggressive kinds you can come across is going to be one that affects the glioblastomas. These are cells within the brain that are meant to support and help the structure of the brain. But because of this, they're fairly well protected from most of what the body can do to deal with them, mostly because attacking your own brain with your immune system is a really bad idea. So cavemen and women who had this trait develop in them were definitely not selected in terms of evolution. This, however, is where a particular antidepressant could come in very helpful. Understand, this is based on the human tissue and mouse studies, so there's a lot of caveats that should apply. So while we say this, just bear in mind it may not play out in the long term. What drug we're looking at here is a particular drug called vortioxetine. It is one of 132 different drugs that were tested in a battery. Understand, this is a fairly, let's say, a basic or rudimentary approach to bioprospecting. You basically take a whole bunch of different compounds and you throw them at problems to see what sticks. In this case, there was 1 out of 2,500 drug responses that they found useful, which is actually quite a good rate. 1 in 10,000 would normally be considered about average, if not slightly above, and that's simply because there are so many chemicals in the world and so many things you might try and test to find one key that matches one lock is literally needle in a haystack. It sounds all well and good, however, there is a fairly big caveat we need to point out other than the aforementioned issues. That is, this is suppressing tumour growth, not necessarily stopping tumour growth, and certainly not making it go away. As such, it's preventing cell division and proliferation, and that does prevent the tumour to a certain extent from invading other parts of the brain and causing other issues, which if you're trying to wait to get in for surgery, it could be an option. But otherwise, this is not going to stop the cancer, it's only going to slow it down, although slowing it down can be just as beneficial in some ways, particularly if the tumour is not operable and you want to give at least some more time to the individual. That some more time can be anywhere up to 30%. 30% may not sound like a lot, but for a person dying, it's basically the rest of their life, so extending it by just that small amount is a big deal. In other news, we have diabetes and possible ways to treat that. Understand, we're talking particularly about type 1 diabetes here, and the proposal is, well, kind of predictable, stem cells. Now, it is very important for us to point out that this has got several elements to it that all come together to lead to our conclusion here. First of all, they're a relatively young woman at 25 years old. They have a type 1 diabetes, but what they did was create her own alpha cells, or insulin-producing cells, by taking her own cells and genetically modifying them and putting them back in. This means that, although yes, they're technically stem cells originally, in the end they became alpha cells, or the insulin-producing cells. The other, let's say, caveat to this is very, well, it's not going to be politically correct, and that is it's Chinese research based on a single individual, effectively a case study. And as such, well, you have to ask a lot of questions about the veracity of it. Something similar happened with, say, the CRISPR babies. They were, let's say, controversial at the time because of CRISPR in humans, but also because the nature of the research meant that we weren't sure if that would actually work the way it was sold. And to an extent it did, but it also raised other problems. In this instance, unlike the CRISPR babies, it's somewhat more, let's say, simplified. That is, rather than trying to make the human body or cell impermeable to something foreign, this is simply telling a cell, don't go down this street, go down this other street, which ends up with the stem cell becoming an alpha cell that produces insulin. It's somewhat more simple. And given that it's just transplanting cells into the pancreas after that, which is a relatively straightforward procedure, this isn't anywhere near as complicated and therefore should theoretically work much more practically. More than that, it's not exactly the first of kind. This has been done before. Admittedly, the prior, also Chinese research, used a male who was 59 years old and had type 2 diabetes, but still, it's the same basic concept, reprogrammed stem cells that were basically made to differentiate into the alpha cells. So this should, if it is as reported, be a likely more common treatment for people with type 1 and possibly even type 2 diabetes. Treating type 1 diabetes is theoretically great. Well, particularly great if you only need to be treated once with this insulin-producing alpha cell, rather than always requiring insulin from an external source. However, stopping diabetes entirely would be even better, and this is where the next research is somewhat more helpful. It's claimed that a bacterial infection could be what's triggering type 1 diabetes. Understand, there are some pretty big caveats to this, not just in terms of, well, it's very nebulous, but also we don't quite understand how type 1 diabetes not necessarily occurs, as we understand that part, but we don't understand the underlying mechanism. Type 1 diabetes more or less requires the human body recognize the alpha cells as being some sort of foreign entity, and as such, they attack them. Of course, given it's part of your own body, that's not good. And more important than that, they're alpha cells, they make insulin. Without insulin, you wind up in a very bad place. So it is a case of autoimmune disease, and most autoimmune diseases, typically at least, are an example of where the body is triggered by some sort of pathogen or some sort of foreign substance, whether it's, say, a pollen causing eventually asthma, some sort of chemical like, say, chlorine causing dermatitis, and there are various other examples. The one fairly common one is where you get infected with a bacteria or a virus and even sometimes fungi or parasites, and they trigger the immune system to be hyperactive. And then through a hyperactive immune system, they just attack everything, including your own cells, which eventually leads them to start continuing to do so, and this just snowballs. Fortunately though, with type 1 diabetes, there's a relatively limited number of alpha cells, and once your body clears them out, you don't have that issue anymore. You have a series of other issues, but at least not that autoimmunity problem. The bacteria the researchers are pointing at specifically as being responsible for this is called Klebsiella oxytoca, and their belief is that this bacteria, when you're infected with it as a young child or baby, is what leads to that particular autoimmune disease, due to bacterial proteins found in the cell wall and membrane of said bacteria. It is, in a very crude manner of speaking, like having a lineup of individuals where you have a Klebsiella and your own alpha cells standing next to each other, and unfortunately, they look close enough that you could pick out either one as being responsible for whatever it is, and so they get charged, or in this case attacked, by your immune system. While it might sound like this is an obvious way to try and protect the body from particularly type 1 diabetes, it's not necessarily the case given how rare the particular overlap between the protein responsible for this and the Klebsiella protein is, with maybe 3% of the population in the UK being, let's say, categorized as having this particular version of it. So, well, that's 3% out of 100% of what could be a diabetic population, and as such, without knowing if this is the only cause, this is one possible cause out of many, or if it's something of a red herring, it's not exactly helpful yet. Understand, yet being a major caveat here, as this could well be a pretty major finding if proven true, and if is the big problem here. In other bacterial news, we have a further, let's say, a herring to chase after, not necessarily a red herring, however, in that some evidence would indicate that a imbalance or particular change in the gut microbiome could be contributing, let's say, to autism and ADHD. Specifically because there's a prediction that if you have a certain spread of microbes, you are more likely to be autistic or have ADHD. Or possibly not have parents who beat you enough as a child, assuming your father didn't go off to buy milk and cigarettes. But no, other than dark humour, there is a reasonable body of evidence that suggests, at least with regards to autism, that the microbiome within your digestive tract does have a contributing, let's say, power. It's certainly not the defining feature, as that seems to be genetic, but it does seem that there may be an interaction, to some extent, between genetics and your GI microbiome, and this eventually leads to some of the autism traits that we see, particularly around fussy eating. Why this research is somewhat important, and we say somewhat as, again, there's a lot of preliminary caveats that come into this, is that by being able to take cord blood, that is, blood from the umbilical cord and not from one of the ancient sleeping ones, you could possibly be able to get an indicator that the child is likely to be autistic. But when this is also matched with similar metabolites and microbes found within the feces of the child, things begin to stack up a little more in favor of this method, as you now have two different but related markers that can be used as a series of tests that can be run on children that are relatively non-invasive. Certainly far less invasive than, say, Wakefield's nonsense with what he did to children and why he got barred from being a doctor for effectively child abuse. But why this is important, again, coming back to the whole point. The markers, and particularly the biomarkers here, can be found in poop and blood. That means you can get a good idea when a child is very young as to whether or not they're likely to be autistic, and if they are, allow you to either do your own research, which is a terrible idea, or preferably engage with those who are professionally trained and capable of providing you with the necessary advice, structure, and support in building an environment more suitable to the child's needs and basically heading off any problems before they become a problem. Now, this can, in theory, lead to another issue entirely around overdiagnoses, but so long as we're operating on the premise of this is a predictive model and you can take steps to help the child before it becomes a more significant issue, or at least minimize any effects from autism or ADHD, you can, in theory, even if you have overdiagnoses, not cause any harm to children who are misdiagnosed, but certainly benefit those who are diagnosed correctly. In other diagnosis news, we have breast cancer. The fact is, breast cancer is getting more and more easy to detect. Not necessarily because it's becoming more obvious, but simply that more people are being screened. This is a good thing. Breast cancer, by the time it gets picked up by a lot of people, is already too late. You are basically on your deathbed, in a manner of speaking, and if you aren't, you will be before long. If you pick it up early enough, you can avoid that, and this is one reason why, although the rate of breast cancer is going up every year, the death rate is going down every year. Barring possibly the USA, where medical care of any kind is often out of anybody's reach, apart from those who are particularly rich. Understand, there is about a 12% risk of having breast cancer if you're a female in the USA, and admittedly, men aren't quite that high, but nonetheless, they have a fairly significant rate of breast cancer, despite having significantly less breast tissue. So this is both good news and bad news. Good news in that, yes, there is more treatment for breast cancer out there, and bad news that there is more breast cancer. In similar related news, we have the interesting article that shows the distribution of immune cells across your body, and it's primarily a simplified explanation of here's the population and here's a very broad part of the body, which isn't necessarily the most helpful, but the image itself tells you a lot. To take a particular quote out of the significance paragraph, our findings show that an average individual's immune system consists of approximately 1.8 trillion cells, weighing about 1.2 kilograms. Understand, 1.2 kilograms of your total body weight is what largely stands between you and death from not only the microbes within your GI tract, but arguably those on your skin, in the air, and even cancerous cells within your own body. It's a fairly amazing feat when you think that, of those cells, even less again are dedicated to those particular purposes, and as such, you actually have a very effective defense system in the form of immunity and your immunological system, with a relatively small amount of your body actually being dedicated to that purpose. Other than the obvious puffery around this, you can see the different kinds and locations of tissues for different kinds of immune cells. And note that T and B cells are all considered part of your adaptive immune system. That is, cells that get better at dealing with a particular thing at any given time. Basically, given enough time, your T and B cells can attack a particular antigen better. The rest of these cells are largely considered part of your innate immune system. Innate basically meaning that it's intended to provide an all-round protection against everything, but it's not the best at dealing with any one particular issue, whether that's a pathogen, a cancer, or anything else. The antigens they can deal with don't even necessarily need to be antigens. For example, macrophages are very good at just gobbling up any foreign matter, whether that's a splinter in your skin, a paper cut, and so on. In terms of paper cuts though, there is a particular thickness that is arguably the worst kind to be able to cut with, and whether or not you use this knowledge for evil or good is up to you. To be short and sharp about it, paper about 65 micrometers or microns thick is the worst kind, and this also happens to correspond with the paper used for dot matrix printers, newspaper, and most magazine and scientific journal papers. So not only now are scientific journals a threat to your wallet, they're also now a threat to you physically. Smugness aside, and particularly poking fun at places like Nature, Elsevier, and so on, the reason for this is rather straightforward. To a certain extent, the thinner the paper, the worse the cut will be, because it's likely to be able to actually make that cut happen. However, there is a range at which you get a diminishing return, and after a certain point that diminishing return goes from being able to make a very thin cut to being able to not even hold its shape when you are cut. Whereas on the other hand, if it gets thicker, you get a wider cut if it can make that happen, or as it gets thicker again, it simply can't make a cut through the skin as the area of contact is too wide. The range that's going to work is between 50 and 100 micrometers, and 65 micrometers being exactly what you want if you want to be a perfectionist, but you basically can work either side of this. The other key thing here is that, arguably, you won't necessarily die from a paper cut, although there are certainly case studies of that happening, and notably, for example, French doctors who travelled overseas got a paper cut and then died from an infection. But for most people, that's not likely to be a major concern. What will be a concern for at least some large corporations will be the, let's say, goal or ambition of 2 Nobel Prize winners who want to cancel their CRISPR patents. Now, depending on how they've patented the technology, this may or may not have effects across the world. For example, if it's a particular kind of patent, even though they've only applied it in Europe, it then gets applied to every other party involved in the TRIPS agreement or something to that effect, wherein every other country now recognizes that patent. And so, if you are relying on something that, say, is licensed in part with that technology, it could mean that you no longer have to pay licensing fees, which is great. It also means that you no longer need to worry about something similar for anything else that relies on the same technology. For example, if you want to modify crops or animals or make a cat girl. The other not-so-great benefit is that it's now free for anyone to use, however they so wish, and that could be a concern. But for the most part, we see this as a net gain at present. That is, some of the limitations based on their patents will no longer be holding people and technology back. To some extent, understand, by no means does this remove all of the patents around CRISPR, just some of them. All of this might sound well and good, and it certainly is to an extent. The problem is that none of these decisions are actually being achieved, let's say, willingly or voluntarily. Instead, it's all part of a much larger interplay of various different lawsuits, patent rights, various authorizing agencies, and so on. And in this case particularly, that those two patents appear to be the focus of a decision that they really can't be upheld. That is, in the long term, those two patents just won't survive. And because of that, unfortunately, they basically decided that it's a choice between a long protracted legal fight they'll probably lose, or making the decision now to simply throw in the towel and say, we give up on those. and they've chosen the honestly more pragmatic solution of simply saying we give up for now. Of course, that isn't the only issue that's being generated because of this particular decision, and we can summarily describe it this way. A number of different businesses have bought licenses for those patents which have now been withdrawn, or at least, denied, and as such, they've paid for something they didn't need to pay for, which does raise concerns around the representation of the various companies responsible for licensing the CRISPR technology, and those two patents in particular. If you're curious as to what it was that led to this particular problem being created, and it's a fair thing to be curious about, it's all down to the nature of patent applications, and particularly in the modern world, how they're getting increasingly more complicated and difficult to submit. What happened was that there was one feature, and understand, it's a single feature of the CRISPR process that wasn't described enough, and in some respects wasn't described at all, in the patent application. And because of that, it's given the general examination of, could a skilled person carry out this claimed method, and the answer was simply no. Because of that, it's considered that the invention was not fully described, enabled, or disclosed, and because of that, it wasn't able to meet the requirements of the patent process. Generally speaking, the idea of a patent is that you give up all of the knowledge around what it is that you have created, but in exchange for that complete, let's say, exposure or revelation of your secrets, you're completely protected from someone else trying to compete with your exact same invention because you have revealed it in its entirety. But that also means that in theory, at the end of the patent period, somebody else, if they have a copy of that patent, should be able to look at it and go, okay, these are all the instructions needed to make this happen. And as you can imagine, given that that's such a small thing to be missing from what is an already substantial patent application, it's likely that the researchers themselves feel rather badly done by. Next we have a example of bad research rather than bad policy application in some respects. That is a paper from 2018 that basically is being pulled mostly because it's a load of crap, and it's all around external key, and the idea that you can treat various ailments from about 7 kilometers away using a, key, which, yeah, we're already calling nonsense on that, but hey, let's assume that through some miraculous mechanism it might actually work. We also would still challenge the idea of it working from 7 kilometers away. The problem with this is simply that, in a manner of speaking, similar to the patent problem, there wasn't a proper explanation of the method. What happened was that the researchers cultured cells and they were given a treatment for 5 minutes. What treatment, you might ask? Who knows? It's not explained in the paper at all. For all we know, they may have simply done a funny dance around the petri plate or a cell culture dish. It's simply unknown. Where this gets ridiculous is that the papers can eventually be traced back to a method which fundamentally involved the lead researcher going into a room, locking the door, and doing something with external key, which sounds a lot like something really shady that should involve TV stars and young children, but nonetheless, the actual process doesn't appear to have any kind of substance. It doesn't explain what's involved in this emission of external key. The problem with this is that it is literally as secretive as young children's television shows were back in the day, and honestly, the results are just as terrible. Death and destruction. Admittedly, at least a different kind of death and destruction, but it does mean that we don't know what the hell's happened, which, again, we go back to TV for you, but also that we don't know what's happened. Who knows what the individual involved actually was doing? Did they have something on hand that they used to poison all those cells? Did they just shake the dish violently? Did they do something else? We don't know what this miracle cure or treatment is, and we're likely to never find out, only that it involves a locked door and secrecy. 2 particular steps, let's say, that are anathema to published research. The point of publishing it is not just to show your findings, but importantly to show how you got to those findings. Results without a methods section are absolutely meaningless. We could tell you that fluffy pink unicorns are responsible for this video, and you would have to believe it based on the degree of quality that these publications have. In other research-related news, we have the US health agencies seeking to bar funding for a particular biologist called David Sabatini, and although this may sound terrible, there is at least some good reason to think that they have not necessarily a right, but at least a justification to do so. Understand, what they're doing this for is based on findings from a sexual misconduct investigation, and arguably, if they're doing so, they should probably, rather than not be able to get grant funding, and simply be fired from their job, which seems like a much more appropriate response if, and understand it's somewhat of a big if, the findings are actually justified. The challenge with this whole situation is partly that there's a lot of ongoing litigation and there's not a lot of clarity and, let's say, ease of understanding of exactly what's happened. There are certainly claims on each side. For example, the institute where he was working claims that it was a improper relationship with a scientist trying to start a laboratory in his institute. On the other hand, he claims that it was a perfectly consensual relationship involving adults who made their own decisions, and that the actual investigation itself carried all sorts of issues around not being impartial. This, of course, means that everybody's accusing everybody else of anything and everything under the sun, and so the whole claim that that report is the basis for later preventing him from getting funding is a big issue. Now, as said, there's a lot of litigation going on at the moment, and as a result of that, we can only really say what's happened in terms of the actual decisions made by the Health and Human Services, but understand that it may not hold up if the litigation on the part of Sabatini does come through as he hopes. Going from the human's small brain, at least the male small brain, to the standard human brain, and why, despite its size, it's not necessarily all advantages, and in fact such a large brain could come with a fairly large cost. When you look at the human brain, it's actually quite large, compared to most of our ape relatives. Chimpanzees, for instance, are significantly smaller, and there is a certain relationship between the size of brains and the size of testicles, with the larger the brain, the smaller the testicles. So it explains why the human's second brain is arguably somewhat more, let's say, persuasive. But assuming that that's not what we're looking at here, the issue is, how does having arguably a more advanced and complex brain come with downsides? After all, you would think that having a brain capable of doing more would be better. However, what has been found, at least according to this research, is that the brain as it is, and particularly the brain structure, because it's so much larger and more complex, doesn't hold up in the long term. It sort of wears out and breaks down in ways that just don't occur, at least by comparison, in chimpanzees and macaques. This does mean that, by comparison at least to our ape relatives, the human brain is far more vulnerable to various issues around degenerative diseases of the brain, and because of that, we have old age problems like dementia and Alzheimer's that, for the most part, apes just don't, or at least our haired apes. The naked ape very much so. And next is astronomical news, and it's news that you should be watching out for. Not because it's actually important in and of itself, but what it means in terms of the events that will occur. On October 1st, the sun let out a huge colossal solar flare, and this colossal solar flare, or coronal mass ejection, is coming towards Earth at a fairly fast rate. Understand, not the speed of light, but still very fast. That does mean from the 3rd of October through the 5th of October, you're likely to see the effects of this coronal mass ejection in the form of auroras. That does mean we should be able to see them very clearly and very brightly in the coming days, ideally anyway. There are various tools you can use, and you'll find some of those linked below, that will give you an idea as to where and how far, either down from the North Pole or up from the South Pole, you can see these. The final news we have for you really just demonstrates the degree of dumb that humanity can exhibit. It's the raid by police on an MRI holding facility because said facility was drawing a lot of power. Understand, there is a good reason why you might want to consider the use of energy when exploring possible drug facilities. After all, grow lights and similar all need lots of energy, it's true. But MRI machines and other radiology machines also require a lot of power. And we mean a lot of power. This is why a raid by the Los Angeles Police Department to try and find the drug facility inside the radiology facility was so silly. Sending in a SWAT team to a room that has a giant magnet in it is going to end badly at the best of times, and in this case, it did end badly. Thankfully, not as bad as it could have been though. What they experienced was simply having the loss of their weapons as it became attached to the giant magnet. Fortunately, no grenades or similar were also happened to be, attracted to the giant magnet. This is all really only known because a lawsuit was filed by the Medical Imaging Center, as basically they're arguing we were wrongly targeted because we use a lot of electricity for our devices that need lots of energy, because they are ridiculously difficult and temperamental devices without that. By powering them up to a huge degree, you get fine levels of detail. If you didn't have that, you'd have a very grainy and bad image. What makes the entire raid kind of ridiculous, other than everything we've mentioned around the problems of using only energy as a justification for the raid, is that the warrant was applied for using the stupid and effectively almost completely, vilified excuse of distinct odor of cannabis plants. You know, the same thing that police use to search cars or at least get a drug dog out to the car? That's the excuse they used. And the fact that they might be able to smell cannabis near there is weird because, well, although it's possible, who would be using cannabis at a radiology clinic? While we won't go through all the details here, just understand, the damage to this is not just limited to the fact they raided the facility, as unfortunately it appears to have been done out of regular business hours. The damages are much more significant in other ways. Mostly that they've chosen to use emergency release on the MRI machine rather than getting someone to power it down, and as such, the MRI machine has lost hundreds of liters of the helium that is used within it to ensure that it works as intended. The same sort of helium we've mentioned in past weeks where it destroyed iPhones. Refilling this helium is not only going to be expensive, but difficult, and more than that, the decision was incredibly unnecessary as it was an emergency release. Losing your rifle to the MRI machine is not an emergency. It's an emergency in how dumb you are and explaining to your field boss how stupid you were to enter the room that said, "Do not come in here with metal objects." But that's not the same thing. That's all the news we have for you this week. If you found it interesting, consider liking, sharing, and subscribing. Please do post any comments, questions, or suggestions you have below